A Christmas Story, Cell Division through Mitosis and Meiosis. The cell cycle is divided into interphase and mitotic phase. Interphase is where the cell spends the majority of its time, and this phase is divided into three parts: G1 phase, S phase, and G2 phase. G1 phase is the first gap phase where cell growth occurs, and then S phase follows and is where the chromosomes duplicate as DNA synthesis occurs. G2 is the last phase of interphase and is where the cell continues to grow and completes preparations for mitosis.
There are 5 phases of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is important because it allows cells to divide and produce new cells so that cells are always replaced. Prophase begins as the chromosomes condense as the chromatin fibers become more tightly coiled, forming discrete chromosomes, and the nucleoli then disappear. Each duplicate chromosome appears as 2 identical sister chromatids joined together at their centromeres by cohesion proteins. The mitotic spindle begins to form, which is composed of the centrosomes and the microtubules that extend from the centrosomes.
In prometaphase, the nuclear envelope fragments and the microtubules extending from each centrosome can now invade the nuclear area. The chromosomes continue to condense and specialized protein structures called connectocores appear on the chromatid and at the centromere. Some of the microtubules then attach to the connectocores.
The cell now goes into metaphase, which is the longest stage of mitosis. The centrosomes are at opposite poles of the cell, and the chromosomes align on the metaphase plate so that the centromeres lie on the metaphase plate.
Anaphase is the next phase of mitosis and is also the shortest phase. Anaphase begins when the cohesion proteins cleave, allowing the sister chromatids from each pair to separate. Each chromatid now becomes a chromosome. The daughter chromosomes begin to move towards the centromeres of the opposite poles of the cell as their connectocore microtubule shortens. The cell elongates as the remaining microtubules that are not attached to the connectocore lengthen. At the end of anaphase, the two ends of the cells have equivalent and complete collections of chromosomes.
During telophase, the 2 daughter nuclei form in the cell and the nuclear envelopes arise. The chromosomes become less condensed, and this completes mitosis. Cytokinesis occurs simultaneously with telophase. In animal cells, cytokinesis involves the formation of a cleavage furrow, which pinches the cell in two to yield 2 daughter cells with equal numbers of diploid chromosomes.
Meiosis is the division of sex cells and is important because it allows organisms to reproduce. Meiosis is preceded by chromosome replication, however, it has 2 cell divisions called Meiosis I and Meiosis II to yield 4 daughter cells with half the amount of chromosomes as the parent cell. This is important because when the sex cells combine, they produce an offspring that is diploid.
In Meiosis I, the original cell will divide to produce 2 daughter cells, each with a diploid set of chromosomes, while in Meiosis II, these 2 daughter cells will divide to yield 2 daughter cells each, containing a haploid set of chromosomes. In prophase I, the chromosomes begin to condense as homologous chromosomes. Crossing over then occurs while they are held tightly together along their lengths, producing new genetic combinations. The chromosomes then move slightly apart, and each chromosome pair has 1 or more chiasmata. The chromosomes begin their movement along the metaphase plate, and the mitotic spindle forms. The nuclear envelope breaks down, and the microtubules from the poles attach to the connectocores, and the poles move toward the metaphase plate.
Metaphase I begins as the pair of homologous chromosomes are arranged along the metaphase plate, with 1 chromosome of each pair facing each pole. Both chromatids are attached to the connectocore microtubules from their respective poles.
In anaphase I, the homologous pairs separate as the proteins responsible for the cohesion of sister chromatids along the chromatin arms cleave. This allows the homologous chromosomes to separate. They then move toward opposite poles, and the spindle apparatus guides them there.
In telophase I, each half cell has a complete set of haploid replicated chromosomes, which are composed of 2 sister chromatids each. Cytokinesis occurs in the same manner as in mitosis, with a cleavage furrow, to form 2 daughter cells that are both haploid to complete Meiosis I.
After Meiosis I, each of the 2 daughter cells produced goes through Meiosis II. Prophase II begins as the spindle apparatus forms. The chromosomes associated at the centromere move toward the metaphase plate.
After prophase II, the chromosomes then position on the metaphase plate for metaphase II.
During anaphase II, the sister chromatids separate and move toward the opposite poles as individual chromosomes.
During telophase II, the nuclei form and the chromosomes begin to become less condensed. Cytokinesis occurs at the end of telophase II, and this causes each cell to split again, resulting in 4 daughter cells with a haploid set of chromosomes. Although mitosis and meiosis both have the same basic idea of the production of daughter cells from one parent cell, they have a few major differences. Mitosis conserves the number of chromosomes set from the original parent cell by producing diploid daughter cells genetically identical to the parent. Meiosis reduces the number and produces 4 cells that are genetically different from each other and their parent cell. Meiosis has 3 events: synapse, crossing over an independent assortment. This takes place in meiosis 1 and does not take place in mitosis. Meiosis has two divisions, first separating homologous chromosomes and sister chromatids. Mitosis only has one division separating sister chromosomes.